Okay, let's get started with the next session. So in this session, we have a wonderful small panel, but talking about some really big problems. We're going to talk about precision medicine and drug discovery and try to work with the audience here and just sort of imagine with you not just what's possible in the future, but what are we doing right now. So let's start with the next slide. Okay, so last year, I promised that within a year we'll have implemented some of these cognitive computing solutions and talk about it. So this year I'm privileged to have some of the systems oncology team here. This is a company that I helped co-found, focused on innovating to transform cancer drug discovery, right? So to be that innovation that's needed to come up with better ways of finding new cancer drugs. And there's Jeff Kiefer and Chris Yu and David Schneider and some of the team from Systems Oncology are here, so I'll introduce you, whoever's interested, to meet them. I'm thankful for some of the slides that they provided for me so I could share with you their approach. And let me start with what Systems Oncology does. So Systems Oncology is a unique business model. It's a unique approach to cancer drug development. It starts with clinical themes, asking a more beautiful question. What is the necessity? Is that working? Okay. So some of the problems with clinical cancer drug development may not be just killing cancer cells. In some cases, the tumor microenvironment and that sort of thing. So asking a more beautiful question, figuring out the research, but I'm going to focus most of my presentation on the knowledge mining and how do you do multi-scalar modeling with machines. So this is the unique part, because when big pharma does this or biotech does this, this is the part that's been missing, and that's the part that we're trying to fill in. And then the back end, the screening and IND ready and all that, that's very traditional, okay? So this front end is the unique part. Okay, so this is all based on hypergraph-based cognitive computing. At the workshop, I spent quite a bit of time talking about the math behind hypergraphs. I won't do that here, but if anybody's interested, we got some publications and other things. But the important take-home message is that this is an evolving knowledge network. You start with machine learning, it learns from data, and you come up with new therapeutic agents that you drive forward. Okay, so that's, in a nutshell, what the whole strategy is. Diving a little deeper, so I want to thank David Schneider for sharing this slide. He leads a team that codes and develops ways of integrating data, but not just integrating data, fusing disparate types of knowledge. And there's a lot of algorithm development that goes into data fusion. And so this slide captures massive amounts of work. Over the last year, there's been a massive increase in the ability of the team at Systems Oncology to integrate any kind of disparate data out there. There's massive amounts of data in the public domain. I'll talk about this, but this is how you capture, store, and represent knowledge is fundamental to machine learning. If you don't do this right, the rest of it is not, okay? Then you need massive infrastructure, and unfortunately, Chris Yu's face is covered by the clock there, but for those of you who are interested, I'll introduce you. He's gonna be here. Chris has developed a phenomenal infrastructure to put all of this knowledge in. So the knowledge graph needs to live somewhere. I'm just surprised with some of these numbers. I mean, if I didn't know that I'd be talking about multi-petaflop systems. 10 years ago, we didn't even use those terms. GPU, 20,000 core GPU systems, it just boggles the mind what's available to us. So we have high performance computing, we have massive data sets, and we're trying to put them all together into these evolving knowledge graphs. So you can break out the process into 3 steps. The first step is you gotta integrate the data. And to integrate the data, you just need really smart coders. These kids, I call them kids because they're like 16 to 18 years old, right? These are young coders, they're real whizzes, and what they do is they write the code that allows us to fuse any data type. In the future, we'll teach machines to do this, but for now, we're using high school students. Why are we using high school students and not graduate students? Because the high school students are outperforming the graduate students. Welcome to the future. These kids are innate in their ability to to use the programming languages. It takes them a week to learn a new programming language, and implementing new ideas is super fast. You still need the PhD level mathematician, so that's Kendall, and she works with a team of mathematicians to develop the algorithms, the math behind diffusion and the machine learning. So that's the second pillar. And then the third pillar, and perhaps the most important, is you need those PhD level systems biologists. That's a picture of Jeff Kiefer. He's in the audience if you want to grab him and pick his brain. He's got deep experience. He's a cancer biologist. He knows about systems biology. That deep domain expert, if you don't have that person to supervise what's happening, then you're building these dumb expert systems, okay? So how does it all work? Well, the first part is autonomous learning. Autonomous learning means the system learns from ingesting data and the experience. So here's a picture of the various disparate data sets. So for example, TCGA stands for the Cancer Genome Atlas, right? So that one source has 10,000 whole cancer genomes. Imagine 10,000 cancer patients, you have complete whole genomes, and you can ingest that into the system. On top of that, you can take the entire yeast genetic pathway and interactions and look for synthetic lethal interactions. You could take pathways, patents, chemical databases, all of this ingested and fused together. So the system learns from data autonomously. The system also learns in a supervised way. So the expert actually curates knowledge directly into the system. And through interaction with the system, the expert could provide new labels, new classifications, and new relationships that don't live in the public domain, but are common sense to an expert. So Jeff is one of several experts who can train systems like this, and I'm using this as one example. And the interaction between the two, it requires a lot of time and effort. So this is a fundamental part of building really good knowledge bases. So, okay, so we have supervised learning, we have unsupervised learning. Does it work, right? So last year, we were just getting set up with experimenting databases like this. So I'm happy to report that we did this over a year now. So when I say we, I mean the team of systems oncologists, the coders, the mathematicians, and experts like Jeff Kiefer. And they started to make incredible discoveries about cancer biology. They discovered new drug targets, new pathways, new mechanisms of action, to the point where systems oncology has now in-licensed 3 new drugs for cancer. One of them is going to the clinic next year. So this is amazing to me because we've been talking about, Anthony and I have been talking about artificial intelligence for years. In healthcare, we'll see this kind of value come down the chain, maybe in 3 years, maybe in 5 years. But in drug discovery, it's here. Drug discovery, AI is already transforming the ability to discover new mechanisms of action, game-changing ideas that could help cancer patients, so we're very excited about that. And what you're gonna hear from the rest of the panel here is AI being used today. So that's part of the take-home message. We're no longer talking about machine learning helping us in the future. It's making a difference today. So with that, let me pass it to the next speaker. Go ahead.
So thank you first so much to Dr. Chang for the opportunity to speak. My name is Allison Callahan. I'm a research scientist at the Stanford Center for Biomedical Informatics Research, and I'm going to talk to you today about the Informatics Consult, which is a new service we're offering to clinicians that is enabling precision in the practice of medicine. Before I go into detail about what the Consult service is, I want to give you some background on why we think such a service is important. And that is that most medical decisions are made without reliable evidence. While we have some 1,700 clinical guidelines across medical specialties publicly available to clinicians to use when they're trying to decide the best treatment for their patient, only about 20% of those guidelines have evidence based on the results of randomized controlled trials. And the reasons for this are several. Randomized controlled trials are expensive and complicated to run. And they also have very rigorous inclusion and exclusion criteria so that by the time you've identified from the possible population of participants those who are eligible to participate in your trial, you actually have very few people. And indeed, they may not look anything like the patient that you have in front of you when you're trying to decide how to treat them. And so what do you do in the case that your patient is very different from that of a randomized clinical trial patient or participant? And moreover, what do you do with the fact that sometimes RCT-based studies are incorrect and wrong, and the results are misinterpreted? So this is where we think that electronic health records can be brought to bear as a rich source of real-world evidence. So electronic health records have both structured and unstructured information. Structured information in the form of diagnosis and billing codes, information about visits and medications prescribed during those visits, and also information about the patient, like their age, their race, their gender, and so on. They also have unstructured information in the form of clinical notes and images, as we've seen discussed throughout the day. And in fact, unstructured data forms the majority of data in EHRs for many cases. In the group I'm part of at Stanford, we use these, or we aggregate and analyze electronic health records data to generate what we call a patient feature matrix, where each row in this case is a patient that we see at Stanford, and each column is a feature about that patient. So the drugs they've been prescribed, the procedures they've been given, the diagnoses for diseases and conditions that they have, and so on over time. And so at Stanford, this is some 2 million patients, 30 million encounters, and this is a real, a rich resource that provides us the opportunity to learn from the experiences of these patients as they've moved through our healthcare system in order to make care better for the next person and enable precision in the practice of medicine. And that brings me to the consult service, our realization of this potential. So the consult service, given a specific case, will provide a report with a descriptive summary of similar patients at Stanford's Clinical Data Warehouse, the common treatment choices made for those patients, and the observed outcomes after those specific treatment choices. And this is an IRB-approved study that we're carrying out over 1 year. And you can learn more about the consult service at greenbutton.stanford.edu. The process involves 4 kinds of people. Requesting physician, so like many of the MDs in the room, an informatics savvy physician, an EHR data specialist, that's me in this case, and a data scientist. And so when a physician requests a consult, our informatics savvy physician works with them to refine the clinical question that motivated their consult request. And once that's done, he passes the clinical question to me, and I generate definitions of the exposures and outcomes that are relevant to the clinical question, and use a search engine that we developed and are in the process of patenting to create an analysis cohort from that clinical question and using those phenotype definitions. We then pass this analysis cohort to our data scientist who does the necessary statistical analyses relevant to the consult. We work together to create a report that then we review with the requesting physician, and that's done by our informatics physicians. So this is our answer to the question of how do you have an interface between a data scientist and a clinician. It's in person and it's a conversation over a report. And the hope is that then the person who requested the consult will use this report in making their clinical decision. And we do this in 24 to 72 hours. So the search engine that we use to generate our analysis cohort is called ATLAS, and it's short for Advanced Temporal Language Aided Search. And it allows you to search and in sub-second time retrieve patient data based on diagnosis and procedure codes, clinical notes, lab results, vital signs, and information about visits, both inpatient and outpatient. And importantly, ATLAS lets you define complex temporal relationships between these features. For example, you can retrieve patients who received a diagnosis of diabetes and within 2 months were treated with a drug like metformin. And this is a view of patient timelines and the points in time in their health record that they met certain features of interest or had certain features of interest. You can see a tutorial on how to use Atlas at this URL. So the consults we've managed so far have fallen into 3 buckets of analysis types: time-to-event analyses, exploratory analyses, which have been the bulk thus far, and inferential analyses. And as an example of each, here's one for time-to-event analysis. In adults with hypertrophic cardiomyopathy treated with beta blockers or calcium channel blockers, is there a difference in the time to atrial fibrillation or heart failure? An example of an exploratory analysis: in patients with incidental interatrial septal aneurysms without other medical problems, what is the risk of thrombus with anticoagulation versus antiplatelet medication? And an example of an inferential question we answered: in patients prescribed ibuprofen, is there any difference in peak blood glucose after treatment compared to patients prescribed acetaminophen? So we see this consult service as part of a broader vision that was first written about in 2014 by Nigam Shah, Chris Longhurst, and Robert Harrington, called the Green Button, where, as I said at the beginning, if you have a clinical situation for which there is a clinical guideline available, we absolutely recommend that you use that level A guideline in making the decision to treat your patient. But if there is not, and as we're finding that's in many cases, we now offer this consult service where you can give us your question. We will use our search engine to find if there are patients that satisfy the criteria relevant to your question and generate a report for you about the outcomes of those patients that can hopefully allow you to make an informed clinical decision. An interesting byproduct of this service is that we have a prioritized list of clinical situations for which clinical guidelines may not be available, and we can actually use that to identify clinical questions that could be studied in further trials, for example. So I want to quickly introduce with faces the consult team. So Saurabh Gombar is our informatics physician. Ken Jung is our data scientist. Vladimir Polony developed the search engine Atlas. Juan Banda manages all things data transformation. And this is really envisioned and conceived by Nigam Shah and supported by Robert Harrington. Our data science collaborators, Robert Tiptrani and Trevor Hasty, and our funding I would like to acknowledge as well. Thank you.
Anthony, it's wonderful to be here. So I want to provide some context to what I consider to be sort of quintessential translational bioinformatics challenge that has incredible opportunity for innovation and precision health, and that is autism. And I'm trying to do that with this particular slide. To achieve personalized health in a complex neurodevelopmental condition like autism, we have to be able to solve this very fundamental equation of genes plus environment equals phenotype. This is the equation that we are attempting to solve in my lab and in the company Cognoa that I helped to found. And it's one that we look at every day as an opportunity space for us to begin to think about how we invent and build new tools and new solutions that can fulfill the parameters of this equation and help us achieve a better understanding of the genotypic and environmental map to the behavioral constellation that manifests as autism. We're doing this by essentially trying to collect 3 modalities of data: the phenomic data, the genomic data, and the environment data. And with those 3 elements, which we believe to be fundamental foundations to achieve subtyping and precision care in autism, we are able to build data fusion methodologies exactly like the kind Spiro was talking about in his talk. And I'll get to that as I walk through this 8-minute presentation. But I also want to say this talk is going to cover briefly some of the touch points that I consider to be opportunities and also solutions currently in play for fulfilling some of these parameters of this G plus E equals P equation for autism. Some of those being mobilized methods of care that simultaneously provide actionable information to the clinician or to the family while capturing data on which we can build better models that personalize. These come in the form of deployed methodologies that fit in smartphones, smartphone devices, as well as in the form of wearables for simultaneous ex-clinical, out of the clinic, home-based intervention strategies that have this dual facet of providing actionable information while simultaneously capturing data on which we can operate using machine learning to build more precision methods. I also intend to describe some of the ways in which we're trying to capture information from the molecular space, the cellular, subcellular space pertinent to autism. It's strongly genetic, it's strongly environmental. We have to get these pieces of information. But how do we do that, and what's the right strategy to acquire the data in a fashion that's at a scale that is appropriate for power, but also in a way that's economical? Autism is a complex condition and represents a public health epidemic today. 1 in 68 kids are impacted. Dr. Sharif Tarman already talked about this earlier this morning. But 1 in 6 kids present with developmental concerns. All of those kids are being pushed to the same clinical standard of care. That clinical standard of care is fundamentally broken. It contains many individuals that probably shouldn't be on that wait list, and those wait lists are growing over time. As a consequence, the average time for getting a diagnostic assessment, which contains this battery of tests, is 13 months in the United States, and the average age of diagnosis is 4.5. That's woefully inadequate. Moreover, on the other side, there's therapeutic bottlenecks. Once the child has been diagnosed and qualifies for reimbursement for care, they're not getting the care that they need at the time they need it, and more, it's a one-to-one relationship between clinician and care provider that also doesn't scale to meet the population demand. And so we have to figure out ways to fix this that also speak to the paradigm that I just described, and that's a lot of what we're working on. One other point about this slide is that the number of questions, number of measurements that are being made within the clinical evaluation of the child is greater than what's necessary. And so you have, instead of a sparse machine learning model, you have a dense one that confuses the clinician and detracts from the actual answer you're trying to get. This is also something Dr. Tarman mentioned this morning in his talk. The more features you're capturing, the further away from the diagnostic quantitative phenotype you're getting, the further away from an accurate answer you're actually getting. Okay, so on this slide, there are 3 kids who have autism. Can anybody find them? This is meant to highlight the important fact that doctors equipped with the empirical knowledge can often walk from their office through the waiting room to the clinical room and hear autism as they're going through. And they're almost always right. This is an incredible intuition that they possess, but they're not acting on this intuition. And so what I want to try to explain in the few minutes of time that I have is that we're building methods of machine learning that mobilize, that enable doctors to get answers quickly, that enable them in turn to act on that intuitive understanding of the child's phenotype that they already have. So in other words, we're going to get them to operate on their intuition in a time frame that accelerates the pace of detection, diagnosis, and delivery of therapy for these kids. We've done this using a series of different machine learning approaches that operate on features extracted from home videos of children, provided directly by the parents in a smart application in a way that's very streamlined, simplistic, low barrier for entry for the parents, but yet provides us with sufficient information in a form of a vector that can be run through a machine learning model to provide not just a very robust answer that, using a model that has a high AUC or has a high ROC, but also one that is quantified, that one that fits a distribution, that tells you something much more important and innately informative than what you can get from just a yes or no. So where they fit on the spectrum. This kind of thing is fundamentally lacking in the field today, but it's the one thing that we need to be able to drive towards precision healthcare and precision medicine in this complex neurodevelopmental condition. An indicator that's coming from a machine learning model like this can put kids on a map in a distribution so you can find kids like mine, patients like mine, parents like me, responders and non-responders, and many other things that are really important for driving successes towards precision medicine. This is exactly what's embodied in a process that Cognoa.com is pursuing today, through which they're getting information from parents in a next clinical fashion, operating on it using machine learning models to present information that is immediately actionable and provides doctors the ability to act on their intuition. To address the other end of the bottleneck that I already described earlier, we've built a system that's a wearable that uses augmented reality to provide automatic emotion feedback to a child with autism, which rescues one of the fundamental deficits that they have, which is facial engagement and emotion recognition. Once that's rescued, these kids will progress off the autism spectrum in many, many cases. However, we're not delivering it in the right way, and we're delivering it too late. We're delivering it using flashcard-based therapy, which don't generalize to the normal the typical population with which the child might engage. For example, if the flashcard has a bearded man representing the angry face, they may naturally correlate beardedness to angriness, and so therefore have a maladaptive outcome. We're trying to rescue that utilizing computer vision technology, in this case deep learning technology, to track faces automatically in the field of view of the child, provide back emotion classification of those faces in real time so that we can give them an actual understanding of what the expression, what the emotion is that's being exhibited on the face with whom they're interacting. This pays dividends. Like I said, this can progress them off the trajectory of autism spectrum and onto a lifelong, typically developing course. Moreover, this feedback loop that is described in this slide encapsulates what I think is critical for precision medicine: the opportunity to deploy remote technologies that simultaneously deliver actionable information, in this case therapy, while capturing data on which we can operate to build better models over time. Imagine the two together. This is exactly what we're thinking about now. The opportunity to get real-time videos from individuals at home in a way that enables us to detect and track, quantify the phenotype over time, while simultaneously providing wearable therapy to the children. But it doesn't address in the 55 seconds the other important part of this equation. That's the P part. I haven't even talked about the G and E. Do I really have to stop in 55 seconds? We have decided that we don't want to wait around for the recruitment efforts that are standard to randomized controlled trials, for example, to get the data that we need at the scale that we need to be able to understand the molecular complexity of autism. So we've begun to develop a mapping system that really borrows from some of the successes from healthmap.org and Flu Near You to simultaneously track the prevalence of autism in real time while getting geographic coded information on every individual and contact information so that we can enroll them into participatory research studies that we run from my lab and from other places across the country. Simply put, we are incentivizing the population of active parents out there to enroll as citizen scientists through this GapMap system by giving them something actionable that they can use in the near term, while also enrolling them in studies that have long-term outcomes and opportunities for moving the needle towards precision medicine. One of the things that we're doing in this category, so basically what we're doing here is utilizing standard channels for marketing and campaigning to build a dashboard for every individual that tells them something important about the nearest resources that are closest to them, while also asking them to enroll in specific studies, one of which I'll describe right now. This is a microbiome-focused study. The gut microbiome is arguably a reasonable, quantifiable environmental barometer that starts to address the E component of the G plus E equals P equation. We've decided that's an important data modality that we have to capture, and we've decided also that we can crowdsource the acquisition of the microbiome in a way that's robust and scalable and economical as well. And so we've done this by asking families with 2 children, one with autism, one without, both 6 or under, within 2 years of each other, to participate in this research study. And we have received an enormous amount of participation from just an internet-based campaign, enough to be able to drive the power needed to identify potential biomarkers that are represented within the environment, which is a significant slice of the pie that explains the phenotypic variation consistent with autism spectrum. And with those data, we've been able not only to crowdsource this, in this case we're just sending out fecal sampling kits to a diverse population of individuals out there in the world, and we're getting information back from sequencing machines that enable us to parse the floral composition of the guts of these children who have autism and differentiate or look to see if there's a differentiation between their floral composition and the floral composition of their sibling who's typically developing. And indeed there is. And so some of these, so the red on top highlights some of the biomarkers we've identified in the autism population. The blue on the bottom are some of the biomarkers that are consistent with the typically developing individuals. What this means to us is that not only are we able to crowdsource the acquisition of a digital phenotype by delivering wearable therapeutic intervention at the same time But we're also able to crowdsource the acquisition of molecular data that otherwise are very complex and challenging to get at a scale that's both powered with the right numbers, but economical enough for us to be able to do it increasingly in the future, so that we can continue to test the hypothesis that these biomarkers are valid. Once we do that, we will be able to be able to build drugs, opportunities for therapeutic intervention in increasingly more robust fashions kind of going forward. So basically, this whole ecosystem. So this is the last one. This is like the most important one. That's my email address on it. Again, this really just encapsulates what I just said. This is just, I think that as we advance towards precision medicine and think about how we can capitalize on artificial intelligence, we have to also think about how we can utilize ubiquitous technologies like wearables and smartphones to capture information in a way that's enticing and incentivizing to those subjects we want to address. But also that capture information that's really that's scientifically valid, and that can enable us to build both labeled training data, to build better models, and deploy those models in a far faster return on investment cycle than what's currently happening in the healthcare system today. So, that's it.
Wow, that was fantastic. Thank you, and congratulations. I think too often you see solutions developed direct to consumer or developments of personalized medicine into clinical workflows. I loved your presentation, loved how you integrated directly into workflows, loved how you brought a solution direct to the families. What's really exciting is there's a research component here, that there's a collective data set that could help in the greater good. And I love that you brought drug development into this as well. You say, okay, we're collecting all this data, we're crowdsourcing, we have different means of collecting the deep phenotyping, the genotyping, and even genomics and microbiome. These are critical features that could feed drug discovery in the future. And the fact that both of you have been thinking about that is wonderful. And so congratulations. Let's talk a little bit about who owns the data. Cognoa is amassing this data. First question.
So obviously every individual owns their data. As they share the data with my lab or with Cognoa, they consent to participate in a way that is comfortable for them. So Cognoa is asking for videos which are entirely privatized and utilized only within the network of Cognoa. On the research side, we ask for more open-ended opportunities to use the information. And in every instance, there's a very robust consent document that they read and they understand, and therefore they all understand kind of what they're getting into when they share this information in the first place. So that's about what
I think that's what a lot of us, I think, unless somebody disagrees, I think that's what we're seeing too in all indications that patients and parents of kids with disease, everybody wants to share the data, everybody wants to make it available to scientists who are doing this type of predictive analytics or drug development. It is a myth that there's an issue with data sharing, and I think it's fantastic that that is being used for good. Allison, there's a couple questions for you. One is, do you mingle external and internal data? And then the second one is, how is Stanford reimbursed or co-reimbursed for the consult?
Thanks. So the first question, temporarily, how is Stanford reimbursed for the consult? So currently, the consult service is supported by an anonymous, generous donor. And we are actively thinking about the question, when this becomes larger, when we want to have a higher volume of consults that we can support when we grow our team, who should reimburse this? Should it be insurance companies? Should it be a service that is purchased by a care provider and we provide service to them? This is an open question, something that we're in the process of trying to answer as we carry out the pilot study. So currently we're not, Stanford is not reimbursed, it's funded. Mingling external data with internal Stanford data, we have commercial claims data that we are also able to use for consult questions, we don't mix data sources. So we will try to answer a question using Stanford data where possible, and if not, or if we need a larger patient sample, we also have access to commercial claims data to ask the consult questions.
Okay, and I'll field one question from me here. It says, Spiro, can you talk about how the systems oncology platform might be used in other settings, neurodegenerative diseases? So the answer is absolutely. We are thinking, so oncology is the test bed for, it's a very generalized knowledge graph. It could be used in any setting. Oncology happens to be one that's near and dear to our heart. We have a terrific drug developer that's working with us, Dan Von Hoff, so that's why I started in oncology. But I could tell you that with Systems Imagination, which is a sister company that has been developing the platform, we are thinking about cardiogenomic intelligence. So Anthony has a passion for cardiology. Why can't we use this to discover and reposition cancer drugs? And I know there's neurology. We're looking at traumatic brain injury with Sharif and Bill Loudon in a company called Neurotherapeutics. Again, it's a graph that brings diseases, genetics, and within each case, what you want to do is you want to take the domain knowledge. So you don't necessarily need TCGA and cancer genomes in this case. What you need is genomes from kids who have had strokes or traumatic brain injuries or cardiovascular disease and so forth. So we're using those, and I think it's a generalizable solution that can help across all indications. So cancer, heart disease, and neurology are the first three, but the sky's the limit. And with that, I want to thank the terrific panel. Very exciting work. Congratulations.